Everyone, so that's our last talk / tutorial of the week, because this afternoon it's all presentations. Again, Claire was very kind to give us a little tour, so I think we're going to dive a little bit into the methods to derive functional connectivity measures, but more importantly, applications.
Yeah, and it's a funny mix, right? Like, so, because I think I covered a lot of stuff on Wednesday, so I'm going to go through some of the more basic methods again, just in case anybody, didn't get it. But I'm going to then highlight maybe some issues, because I like to do that.
And that's a technique with lots of issues, so lots of material. All right, stage is yours.
Okay, but so I thought I said that I would say a little bit about my career path or whatever, in case. I suppose people at the beginning of their careers are often interested to hear how people end up where they are. So you all know I'm from Ireland. I'm from this dot up there in the northwest, pretty close to the border with Northern Ireland, which, as you know, is becoming a bit of an issue again. And I went at 17 to Trinity College. And did my undergrad there in psychology, then PhD, again in the psychology department, but I started working with Hugh Garavan. Some of you might know he's now a professor actually in the University of Vermont, and he is one of the, I don't want to get his title wrong, but he's got quite a big role in the ABCD, the Adolescent Brain and Cognitive Development Project, that huge data collection project going on in the US at the moment, involving data being collected from over 10,000 kids who are going to be followed through adolescence and into adulthood. So that's where I started to learn about FMRI. I started, what I was interested in for my PhD was practice effects and how practice and learning change patterns of brain activity. But after I finished my PhD, I went to New York, and I suppose, as most people do, I went thinking I'll be there for maybe 2 years, do a nice postdoc, and move back to Ireland. As these things go, I just ended up staying there for a really long time. So I was there for pretty much 10 years before I went back, decided to go back to Ireland. So it was at the Child Study Centre where I worked, at NYU Child Study Centre, where I worked as part of this group with Mike Milham, whose name I'm sure many of you recognize, Javier Castellanos, who is very, very famous a child psychiatrist with lots of expertise in the brain bases of ADHD. And we really had this kind of, this time that I've been talking about the last few days, is this like golden period. These periods are always only evident in retrospect. You can only tell it was a golden period because it ends. But we had this period of just huge productivity. We were at the beginning of, I suppose, maybe the second wave of interest in resting state and what it could tell us about the brain, about brain functional organization, about its development, about its disruption in psychiatric disorders. And so we produced a lot of papers at that time that ultimately have become very, very highly cited. And so that has been enormously helpful. It makes my CV and citation indices look really, really good. But I think more importantly, it was really, that was where my career was forged, my interests, my abilities, my skills, and so on. And it was it's a really lovely time to look back on in my career. But of course, things change, people move on, people change institutes, and the time came for me to decide, was I going to settle down in the US forever, or return to Ireland? And first this one came along, and later, after I moved back, a second kiddo. And those, I suppose, helped, push me back towards home. And I think that often happens for people. Yeah?
Is that child in an MRI ward?
I actually used this picture in my talk the other day. So he wasn't in the board, but he is a model so Adriana Di Martino, whom I worked with at NYU, but she's now at the Child Mind Institute, does amazing work on autism, and she's particularly interested in looking at the brain in toddlers who have autism, because she really wants to try and, because it's such an early arising disorder, she wants to be able to image them as early as possible. So she has been doing amazing studies, sleep studies, where she gets autistic and non-autistic toddlers to sleep in the MRI. And so Donica was being a model for photographs for, I suppose, a brochure that explains to parents and kids the procedures involved in the study. And I think she still uses it. So, and being his kind of typically contrary self, he wasn't particularly compliant. So we never actually got him to, this was just a mock scanner. And we wanted to get him lying down, but we never managed that. So I was trying to think, like, what exactly do I want to kind of give you to take home about my career path? Because I suppose I've been very, very lucky, and some things people often, especially at home, they often highlight my H index as something that illustrates that I'm an amazing scientist. So a lot of that is luck. We worked in an area that became very, very popular. And so many of my early studies, because they were quite foundational studies talking about reliability, establishing the initial validity of the functional connectivity approach, they have been really, really highly cited. And that might not have happened. And loads of people, loads of amazing scientists don't have H indices of whatever, 50. It's something that not to get hung up on. And I think these days people are there's a shift away from those kinds of measures of importance or contribution or whatever, towards the kinds of things that you are doing here. So the kinds of skills that you're developing, the practices that you're literally, learning to do and doing as part of your own projects. And those are increasingly going to be how scientific productivity and importance and impact are measured. We're going to start reporting, how many times your data set or analytic scripts or whatever they are, how many times they were downloaded, not how many times your paper, wherever it was cited, So I think it's a really exciting time with lots of change. And I think part of the change can be scary, because the world seems very competitive. It seems, particularly this area of functional connectivity now is so big. There's loads of people working on it, but I think it's also a real opportunity to push the field towards these, the fair principles and really good, solid science. Okay, so I'll get off my soapbox. I'm just going to go, like I said, I talked through a lot of this stuff the other day, so I'm going to go really quickly through just the basics of functional connectivity again, and then I'll move on to just highlighting some of the things that we looked at in our earlier work, but also where I think there's a lot of work left to be done. I'm sure you're already familiar with this, and I did already go through it the other day, how we measure task evoked activation with FMRI. At its simplest, we're just looking for where there are changes in activity in the brain that map on to some cognitive task that we have imposed, or some differences between task conditions and so on. And so at its most basic, we contrast, times when we're giving somebody tasks versus times when they're not, or we contrast, a difficult condition with a less difficult condition and so on. And it really is quite a simple, straightforward analysis that gives us these maps. And how it's often implemented is through this general linear model say something like FSL Feet is one of the packages that's often used for task-based MRI, but also for functional connectivity, because at the end of the day, the main difference between a functional connectivity analysis, particularly with resting state data, and a task-based analysis is that rather than having this design matrix that codes the events of interest or the changes of interest in the task that we want to pick up on in our analysis, we just have a time series that's derived from the data itself, right? So in this case, we have our pre-processed task data and we have our task design. We run this GLM and we get a statistical map of what voxels in the brain show changes in activity that correspond well with our design matrix. In resting state, we're doing the same thing, but just with a time series of data taken from some region of interest at its most basic. Okay, so I'm just going to skip over this stuff because this is what we talked about the other day. I highlighted the importance of why it's useful, why it's interesting to look at functional connectivity. And I think I kind of touched on all of these issues, like why task-based fMRI is not a tenable approach when it comes to working with developmental populations or animal populations. There are real challenges to even if we can get kids to lie still in the scanner and perform some sort of cognitive task that we're interested in, there are real challenges to kind of getting the task right, equivalently challenging both the children and, say, the adolescents or the adults cognitively by that task. Because if we don't know that, if the difference between a child's performance and an older child or adult's performance is just a difficulty effect, then that's not telling us anything really about how the brain areas supporting task performance develop over time, right? So if there's just kind of a big task difficulty effect, or there could be a ceiling effect where adults just find the task really, really easy and aren't really engaged with it. So these are many of the reasons why I have kind of seized on this resting state approach as something that can give us those insights into brain function across development. Okay, so what this is a really simple representation, right, of what the data look like on a volume by volume basis. So with functional MRI, right, we're collecting a whole brain, three-dimensional functional volume at every TR, every repetition point. And so often this is 2 seconds. These days it can be less than that because we've got much better acquisition methods, but the standard for a lot of functional connectivity analyses for a long time was just 2 seconds. So we have a whole brain volume that captures that bold contrast, right? This kind of proxy of neuronal activity that we use. And just taking like one single voxel, this is a representation of what the time series for that one voxel in one brain area looks like across the duration of that scan. And obviously this is a cartoonish representation because with unprocessed data, it's a whole lot noisier than that. But this is actually what it does look like after all the pre-processing that we apply. And as I mentioned the other day, I don't think I showed this slide, but to compute functional connectivity, all we do is we take one of those time courses or an average of them across a region of interest. So we have this seed time series, and we ask what other voxels in the brain show a pattern of activity across that resting state or movie or whatever state it was that the participant was in, show a pattern of activity that's correlated with my starting region, with my seed region. And so it really is as simple as a correlation analysis, at least the first step of computing functional connectivity. So you can do that by loading in a pre-processed volume to MATLAB or using Python, whatever you want to do, and computing correlation across all the voxels in the brain, because that's functional connectivity. It's just a measure of correlation, and that's its most simple, univariate, really the absolute vanilla kind of way of computing functional connectivity. And there are many, many other ways, and more sophisticated ways, and much more multivariate ways. There are things like independent components analysis, ICA, that I'm sure you'll have heard about and talked about. But essentially they're doing the same thing, right? They're just detecting patterns of correlated activity, but they're doing it in a more data-driven way. They're not, with less kind of top-down input from the user. So by doing this, by running this simple correlation analysis, we get, a map of functional connectivity. Correlation values is R values. These are often just then converted to, you do a Fisher R to Z transform before group level analyses, which again are just exactly the same as the kinds of group level analyses you perform with task-based imaging data. So these days, that's typically some sort of non-parametric permutation-based approach, for example, randomize with FSL, but there are lots of different ways of doing that. But you're doing the same thing. You're taking statistical maps from individuals and performing some sort of group-level statistical analysis on those and applying appropriate corrections. So I showed you this, the original, Bharat Biswal's very first demonstration of functional connectivity, and I think you're all. This is probably just almost accepted canon at this point that that functional connectivity analyses of resting state data do reproduce these patterns of task-related activity really faithfully, and so we can pretty much obtain any task-related network that has been obtained with task-based functional connectivity, task-based FMRI, using functional connectivity analyses that focus on the regions that were that either are data or hypothesis-driven and look at the connectivity associated with those regions, like Bharat Biswal did in his original study, or that use more data-driven approaches like ICA, which was the case in this Smith paper from 2009. So, are there any questions or any observations or anything at this point? Is it all fairly?
Maybe explain a little bit what the brain map.
The brain. Yeah. So sorry, I didn't explain it. What they're comparing.
People actually don't know about that stuff.
Yeah, and I suppose brain map, in some ways, has been a little bit superseded by things like neurosynth, right?
They have pros and cons.
Yeah, so brain map was something that was originally written by Angie Laird and is it Peter Fox?
Fox.
Yeah, Peter Fox. So yeah, back in the earlier days of where things were very much still focused on task-based fMRI analyses, people would often do meta-analyses, and I did present a slide on that the other day where I showed how the patterns of functional connectivity, for example, associated with a seed placed in Broca's area, mapped on really well to the pattern of activations seen in a meta-analysis that combined, that used coordinate-based meta-analysis, right? So where the researchers are taking whatever studies they can find on a particular task or functional domain, so in that case it was lexical processing, and they go through all of the published studies and they pull the tables that report the peak coordinates of activation for that task. And then they perform a statistical analysis on that. They can use, you could use a brain map, right? There was, what's it called? There's a coordinate-based meta-analysis program.
There's ALE?
ALE, that's it, yeah. And so you can enter those coordinates and you can perform this coordinate-based meta-analysis, which gives you, where across the brain activity was most likely to be seen across these different studies. And then things like BrainMap did that on a large scale, so they pulled, I think you had to enter your coordinates right from your published paper. I don't know if they pulled it automatically, did they?
No, they don't. They're manually curated, and
Yeah.
I mean that resource still exists.
Yeah.
I think it's complementary to neurosynth.
Yeah, I saw Neurosynth, and there's another, I did want to mention Neurosynth, but there's another one that people talked about at HBM that I thought was based here somewhere, in Montreal or in Canada.
Not that I'm aware. But I mean, I just maybe like to explain what those
To explain what these maps are.
In terms of the meta-analysis, if some people are interested in meta-analysis, and Giller gave a talk at New Academy like 2 weeks ago, and it's been recorded, so I can post a link if you want, because she gives an overview pretty systematically of like the recent evolution. But just like because you didn't explain
what the measure of task-based activity was. So it was also, so these were measures of maps, functional connectivity maps derived from ICA, applied to I think 20 participants' resting state data, and then these were also ICA applied to. These are derived from ICA applied to the BrainMap derived maps of task-related activation associated with many different functional domains. And so what Smith et al. Was showing were that you could obtain the same kinds of maps across these two different modalities. These were quite different modalities, and at the time this was a very compelling demonstration of the validity of functional connectivity. I think now it's almost taken for granted, but yeah. So yeah, I think people tend to use maybe Neurosynth a bit more, and yeah, I couldn't remember. There is a newer version of Neurosynth, but Tal Yarkoni is not involved in this one.
Maybe you're thinking about NeuroVault.
No, it's something else altogether, and I'll try and find it or remember what it was. I think they were arguing that their way of pulling the information from the published studies was better or something, but I'm not providing a good summary of that right now. Okay. So I thought I would just touch on one of the other. So most of my work focused on just that really simple kinds of functional connectivity analyses. A lot of really the seed-based approaches. We also used ICA quite a bit, but that's at about the level of sophistication that I got in a lot of my papers. But one of the other approaches that we use quite a bit, because Sinyan Zhou, that some of you may know, was a postdoc in our lab at the time, and he brought with him this method that he had developed with colleagues in China called ALFF, which Javier always kind of bemoaned that was not a very good, not a very nice abbreviation, doesn't roll off your tongue. So it stands for amplitude of low frequency fluctuations. And it's actually just a measure of the activity. So rather than focusing on connectivity now, you're just looking at the time series itself, or the brain activity itself, and quantifying that. And essentially what it is, you pre-process the data, you apply an FFT, and so you get this power the power spectrum, and you quantify the amplitude of the power in particular frequency bands. So for ALFF, it was just the sum of all amplitudes between 0.01 and 0.1 hertz. And it's analogous really to taking the standard deviation of the time series. It's what a lot of people have started to talk about now. Like, people have become very interested in the variability of the signal, but this is exactly what ALFF captures as well. And so people have been looking at this for quite a while, just under different names. So Randy McIntosh is in Toronto, is somebody who's done a lot of work on variability of the fMRI signal and the resting state signal, particularly with relevance for aging. So people have been doing this for quite a long time, and so Sinyan, one of his earliest papers in our group, was looking, was just kind of characterizing this in a more comprehensive way, this ALFF measure, and then looking at its reliability. So you can see that it's really very much localized to grey matter, for example. There's this fractional one that is a ratio of the power in that specific kind of low frequency band relative to power outside it, and so on. They're pretty similar. So this is, I just wanted to highlight this as another potential measure that can be derived from, another very simple measure that can be derived from resting state or movie data or whatever. And it's one that maybe hasn't been explored as much as it could be, and that's part of the renewed interest in it. I'm not sure how useful this is. I'm going to just talk very a little bit about the pre-processing, the steps involved in, deriving this very simple or these measures of functional connectivity. But there are so many resources out there, and I'm sure you've kind of covered a lot of this already. And there's also, I suppose, constantly ongoing debate in the field about what are the best pre-processing steps. What is the best order, the effects of using different orders, and all of these things do seem to have some effect. I think it's debatable how much. Within limits, most people apply this kind of central set of pre-processing steps. So stuff like slice time correction, actually I don't do that anymore, and most people probably don't do it for resting state. What happens with multiband? You do have to do something there, or?
I mean, multiband is a recent type of acquisition where you acquire multiple slices at the same time. So it creates all kinds of artifacts.
Yeah.
But it brings your acquisition time very low. So slice timing correction tries to correct for the slight differences in acquisition times between different slices, but because the acquisition is much faster, you don't really care about it as much. So I would say, it's not really
not commonly done, yeah.
With the multiband. Now,
yeah.
Basil has worked on ways to correct for motion within a volume, actually leveraging the fact that you have multiple slices acquired at the same time. So I would say that's kind of like for that top level set of corrections to be a multiband specific thing.
Yeah.
But I don't think at this stage it's commonly applied by anyone. It's very much research direction. Yeah. So it's always really interesting to me how papers continue to emerge, focusing on some of these things that have just been ongoing for years and years and years. Like, what are the best set of pre-processing steps? So slice time correction is something that most people probably don't do. Obviously, motion correction is very important. Despiking is another type of detection of extreme motion or other time points that are associated with a big jump in signal for some reason. There are lots of different ways of addressing those. So despiking is one, so it just kind of squashes those extreme time points. Grammean scaling is scaling all of the data to have the same mean, which is necessary because of our FMRI, not of it being a relative measure rather than an absolute measure. And linear and quadratic detrending, which is, and maybe people do even more kinds of detrending with longer time series, but again, those are just to remove drifts and other nuisance signals in the data. But where a lot of people have dedicated a lot of effort is this this middle area of nuisance signal regression, and I'm sure maybe some of you will have heard about all of the controversy, first of all to do with something like
something called global signal regression, which is where the pre-processed data are regressed on a global mean. There's just, I don't know how many papers on this issue, and it's controversial because it does mathematically force the data to force measures of functional connectivity to contain both negative and positive correlations. So that's a mathematical fact that's easily demonstrated. So what's disagreed upon is whether that's a meaningful thing to do or not, whether it's the right thing to do or not. So a lot of people argue that it does improve, say, the anatomical validity of the measures of functional connectivity that you get, whereas others have argued and shown that it's problematic for example, group comparisons. The global signal probably contains meaningful neural information, but it also clearly contains lots of noise, so physiological noise, motion. So papers looking at the how to properly address the problem of motion and functional connectivity data have really, I think, convincingly shown that that global signal regression is pretty much the best approach for getting rid of motion artifact from your data. So I don't want to dwell too much on it, just to say that that's, again, an ongoing controversy. But people like Ted Satterthwaite in Philadelphia, UPenn, has published some really nice papers looking at these different, or trying to optimize the nuisance signal regression to address motion issues, particularly in developmental cohorts. And so they suggest a kind of a combination of global signal and these other nuisance signals derived from white matter, CSF, and then motion parameters. So you may remember, if you talked about motion correction, that you get these 6 parameters that correspond to displacement in 3 directions and rotation in 3 directions. And so what people typically do is they take those 6 parameters, they take their first derivatives, they take the squares of the parameters and the squares of the first derivatives, and these, along with the same kinds of transformations of global signal, CSF and white matter, and then regress the pre-processed data on all of those nuisance signals. There's concerns about how many degrees of freedom you lose with these kinds of very comprehensive nuisance signal regression approaches, but they do seem to, it does seem to be necessary to address some of the motion issues. And we know that motion, even very small motion, is quite impactful for functional connectivity, particularly if you're dealing with, say, developmental cohorts where you know you have more motion in one group, or you have, a correlation between motion and whatever your behavioral variable of interest is. So once we have this nice, so I suppose something else that's worth saying is that this is maybe one way of doing it, like where you regress your pre-processed data on these nuisance variables, and then you take the residuals of that regression. That's kind of your cleaned pre-processed data, and then you can do your functional connectivity analyses on that. But there are other ways, and I think that, Pierre, you probably do. Certainly the tutorials, I have I'll show the tutorials in a second, do the connectivity and the nuisance stuff at the same time. Yep. So this isn't the only way to do it. This is just one way that I tend to do it, because then I get this clean data and I can just work with that and I forget about all the confounds.
So yeah, I think, that's more like a logistic sort of choice.
Yeah.
Whether you produce all your maps and then you do your modeling on it, or you go from the sort of minimally preprocessed data, essentially almost like starting from the blue, and on the fly you're going to regress and smooth. And I think like with NiLearn has more this approach where you do the preprocessing, the regression on the fly, and the reason is that stuff is fast, and then you can easily test multiple choices, because all those controversies if you want to do global signal regression or not global signal regression, if you start from minimally preprocessed data, it's very easy to test both with the same script.
So that didn't used to be the case, right?
Yeah.
Like, so when we started doing this, it used to take a really long time.
Yeah.
And I think that's another one of the big changes, like how quickly these things can be done.
It's still very slow. So minimally preprocessed data is what FMRI plug generates, or HCP pipelines also have minimally preprocessed data. And basically what they do is all the normalization, nonlinear transformation, which is in the green, is like freakishly slow. But depending on the tool you use, if you want to go to surface with FreeSurfer, we can talk like 6 hours per subject, like something crazy like that. And but the regressions themselves are
that's probably pretty fast. Yeah.
So that's what Nylearn does.
Yeah, and I suppose that's where we would try to save time, so we would do all this, and then we would take that this is all done in native space, in something like 3 by 3 by 3 millimeter, or whatever your acquisition size was, and then we transform that pre-processed volume to standard space, and then we just store that, and use that as the basis for computing functional connectivity, or running ICA, or doing whatever it was we were going to do with the data, so that we didn't have to do this on the fly each time, but it, perhaps not as necessary these days. So this is kind of then your fully pre-processed volume, and then you can do whatever approach you're going to do using that before running group analyses. Yeah.
Can you describe a little more the time series extraction part of it, such as this term seed is new to me?
Yeah, sure. So seed is just a word to describe the region that you're extracting the time series from. I suppose like people don't do it quite as often anymore. I think it's a really, not to say that you shouldn't, but so you can either use an anatomical region of interest, or you can use some sort of mask, right? Do you know what I'm talking about when I say, a mask that just will extract the time series from a specific number of voxels, and you can either base that on anatomy or task activation, or whatever it is, a map from Neurosynth, or you can create, what we often used to do was create these spherical regions of interest, just using a program in AFNI to create little spheres, and then you apply that mask to your pre-processed data, usually in standard space, and what you get is just an average time series, or you can do fancier things. You can do a PCA or something on all of the voxels that are in that region of interest. But at its most basic, we just take the average, and then we just get that time series to run a correlation with. Does that make sense?
Yeah. Also, is there any cross correlation that is done automatically to see, like if there are two areas that are kind of more far apart?
Yeah.
To see if the signals match up.
No, so I think people probably did some analyses like that, and then there has been a bit of controversy associated with, particularly earlier on, people were really interested in applying things like Granger causality analysis, so trying to figure out whether there was directionality in the interaction. So whether we could say, prefrontal cortex, the connection between prefrontal cortex and parietal cortex is such that there's seems to be information flow maybe from prefrontal to parietal or something, and they do that with things like Granger causality, which look at the time lag, I suppose, or differences in the time series, and that has been quite controversial with FMRI because the method doesn't permit those kinds of inferences certainly in the context of resting state, because there's variability across the brain in the hemodynamic signal, in the hemodynamic response, that could drive those kinds of causal inferences in a completely spurious way. So I think it's permissible in the context of possibly permissible in the context of tasks, but not in resting state, even though I have a paper that did it. So I'll kind of unfortunately have to say that's not, you know, a good, and I haven't really seen any analyses recently that use it.
So actually, I mean, I would say it's not controversial anymore. It was shut down, basically.
Well, I had to review a paper recently that did it.
Yeah, so Steve Smith, who published that 2009 study on ICA, went on a crusade for a few years. He did this beautiful simulation study where he demonstrated, like Claire said, that the different delays in hemodynamic response would be actually larger than the neuronal delays, and that grandeur causality could just not be estimated from FMRI time series. And there's still people using it, but I would say they've been cornered.
Yeah. No, well, I blocked that study, you know, like for those I mean, you can just say sorry, but people have pretty comprehensively demonstrated this isn't permissible. So I think that kind of cross-correlation thing is, you know, would fall under the same problems, right? So, okay, so I wanted to post this because this is what a pipeline used to look like. These were scripts that essentially came from, you know, my pipeline that I created when I started working with these data first, that were ultimately shared and kind of became the basis of what a lot of people around the world started using for functional connectivity analyses because of this 1000 functional connectomes project. And I have to say that I'm pretty embarrassed to admit that this is essentially what I still use. I haven't yet made the full transition to a reproducible pipeline, which I should. Yeah?
Oh, I was just wondering if you were feeling lethargic today at 5:01 p.m.
You know, my clock is still on. It must be still on.
Or sorry, nostalgic, not
lethargic. What's that? The.
March.
Oh, yeah. Why is that? Oh no. Do you know what? This is an old screenshot. Yeah. It's not. Yeah. So I think I took this a couple of years ago. Yeah, so this is kind of where I still am, unfortunately, but I'm hoping to change that in the next couple of months. But these scripts were, you know, these were kind of some of the first ones to be shared openly in the community, along with this huge and first really big data sharing effort, like public, you know, making data freely available with no strings attached, no you have to include me as a co-author on your papers or whatever. And it's kind of, I think it's probably a little bit hard to appreciate like what a watershed moment this paper and Mike Milham, who's my postdoc mentor, like what those efforts have meant for the field. I mean, that they were the first data to be shared, right, Pierre? It's not.
Well, at this scale, for sure.
Yeah.
There's no question.
But even Open FMRI didn't exist till 2011, I think.
If it did, that was Task FMRI, and there were very few data sets on there. So that data set was like 35 sites, 1,000 subjects, 1,200 actually, something like that.
Yep. And it was really, like, it really was groundbreaking, you know, I think it's, you know, I don't mean to say, oh, you young people don't appreciate things these days, but I think it is kind of hard to appreciate, like, what it was like before that. So if you were a small lab with no resources, who found it hard to get grant funding, it was hard for you to do neuroimaging research, right? Because there weren't these data sets that you could just download and work with. Now, anyone in the world, as long as they have some computational resources, can get their hands on data, work on it, and write a paper and publish it, and do neuroimaging research, even if they're not well funded. And it's funny because so I call this like data socialism. It's like science socialism. And Mike is not a socialist, but he is when it comes to data. So you know, I think it is worth highlighting like how important this stuff was. But yeah, just to I wanted to draw your attention to this because actually I hadn't seen it and Pierre showed it to me the other day. This is an amazing set of resources that go into way more detail and with hands-on examples that you can actually reproduce yourself, and doing all of the things that I just talked about. So extracting signals, like using a brain parcellation, so that's instead of using one specific seed, but actually taking a whole brain parcellation or a parcellation of an area, extracting the time series from your pre-processed data and computing your connectivity matrix. Here's spheres from an atlas, like many people produce these. So there are parcellations like the ones that Pierre has worked on, atlases like, what's his name, Jonathan Power. So a lot of the WashU folks have produced these kind of atlases. Then Thomas Yao has produced lots, so I have some of those. I'm going to skip over all this stuff because I want to talk about the
By the way, the nylon tutorial pulled a lot from the data you were mentioning from ED.
Oh, really? Yeah.
Yeah, DT200 and Abide, pre-processed in particular, are the basis of a lot of those examples.
Yeah. So, and I think that I suppose it's easy enough for me to say, oh, it's very straightforward and very simple. But with those tutorials, you can just, you could literally reproduce what I'm saying in a matter of minutes and get a real sense for what these data look like and what are the outputs that you can get from them. I'll just, because I'm already at an hour, so I'll just mention this really briefly. Like this is something that Pierre is an expert in, this data-driven parcellation. So using measures of functional connectivity to determine, I suppose, the building blocks of the brain. So by computing functional connectivity, so here I'm just doing it for a bunch of voxels located in the insula. So I compute the functional connectivity of each one of those voxels across the whole brain, and I get this matrix. But then then I compute the similarity, between the whole brain patterns of connectivity for each of those voxels. And I get this matrix, and then I can apply some sort of clustering method to group voxels together that have similar patterns of functional connectivity, right? So it's just another way of looking for regions in the brain that are, we could consider them functionally distinct because they show, so within a cluster we have similar patterns of connectivity, and then between clusters we have different patterns of connectivity. And so this has been a really commonly used approach to detect this kind of overall functional architecture of the brain. So between these kind of maybe large scale networks, like Thomas Yao's 7 network solution, to much more fine-grained ones like from the Schaefer Atlas in 2018, or Sebastian and Pierre's paper that some dreadful reviewer did not complete their reviews on until last night.
You did?
I did. But this, so using this approach to try and detect like what are the individual units in the brain that differ in terms of their functional, that differ or are the same. So you can kind of build up from these like really atomistic, like really small regions of regions defined in this way, and they can be kind of built together into these bigger networks, like kind of 7 networks as opposed to 444. So showing this, that they show this hierarchical organization that tells us about how brain function is organized. And this was so just to highlight this one paper where I was using that kind of approach and more to further validate what we're getting when we apply this kind of approach to functional connectivity. So I applied it to task coactivation, which was from the brain map patterns of task-related coactivation from brain map, but also structural covariance. So similarity in gray matter volumes across individuals, and showed that could get really similar parcellations on the basis of all of those kinds of data. Okay, I'm just going to talk about one thing really quickly. Daniel's stuff is awesome and you should check it out if you have time, and I'm not going to try and explain it. But some of the work that we did was looking at reliability, and this is something that continues to be a big issue and big question in the field. So we had some of the very initial papers showing that measures of functional connectivity were reliable, and the ALFF, so measures of the activity itself, were reliable, and ICA-based measures were highly reliable within adult samples over timescales from within a session to about a week to about a year or a year and a half. Excuse me, but Sinyan Zoa has really taken this as something that he's continued to focus on. So he has a lab. He's in Beijing, right? Sinyan?
Somewhere in China.
Somewhere in China. That's not. I think he's in Beijing. But so this just came out, I think, last week, and it's a really nice commentary in Nature Human Behavior. Again, highlighting how important reliability is for neuroimaging research. And so one of the things that has become really clear is that, and we know this across the board, right, from behavior to neuroimaging to whatever measure it is we're looking at, the samples that we have been typically working with in the field are just not big enough, or haven't been big enough. And recently people have realized that we need to have bigger and bigger and bigger samples to have meaningful measures to really ensure reproducibility, to have adequate statistical power to detect things and to have reproducibility. And this has been, this is a challenge, right, for neuroimaging, because if you're a small independent lab like me, it's hard to get, to collect several hundred participants, and so the way most of us can obtain those kinds of samples is through the data sharing efforts that exist. But one way, and this is what this paper highlights, one way that we can reduce the sample sizes required is by increasing the reliability of the measures that we use. So that's one of the things that I'm kind of quite interested in trying to do in my lab is, and I talked about this, like one of the ways we can do it is by, well, I didn't talk about it too much the other day, but one of the ways we can do it is by trying to figure out, like, what are the best measures of functional connectivity? How can we improve, so how can we make those measures more reliable? And one issue that I think hasn't really been addressed very well in the field is that we see lower reliability in kids relative to adults, and we see lower reliability in neuropsychiatric disorders relative to typical individuals. And this is then a big problem for reproducibility. Because if our measures are more unreliable, we really do need these enormous samples in order to detect reproducible differences. And I think there are some kind of null studies being published, so I think Lucina Udin, for example, published a study in Neuroimage just last year where they were looking at differences between kids with autism, ADHD, and typical controls, and they didn't get any statistically significant differences in relatively large samples. And that's a bit of a worry. And I think part of that is that our measures of functional, are kind of, at least these plain vanilla measures of functional connectivity, as they stand, are perhaps not as reliable as they should be. So I just wanted to highlight one or two things in the last couple of minutes that I think are interesting and worth pursuing in this area to try and improve our reliability, or try and figure out what are the best ways to quantify that reliability, right? So Mike Milham at Child Mind Institute still publishes a lot of work, and Sinyan publishes a lot of work on reliability. And they often use this measure called ICC, this interclass correlation coefficient, that I don't really like very much. And I think that this method from Emily Finn, who is in Yale, but is now...
Prof.
Yeah.
If you want to get a great postdoc with somebody fantastic, at Dartmouth.
At where?
Dartmouth.
Oh, Dartmouth. Yeah. So she just got a faculty
school.
position there.
Yeah.
So definitely. I would say she would be a great person to work with. And so she's developed this really nice fingerprinting method, which is just quite simple, where you look at how well an individual's kind of just connectivity network for a particular set of regions or seeds or regions of interest or parcellation or whatever, how well their connectivity network identifies them from a bunch of other people using a second set of scans from the same group. And what they find is that, again, in adults, your connectivity fingerprint, your patterns of connectivity can identify you quite well, very well, from amongst a bunch of other people. But again, that doesn't work quite as well in kids, and I'd really like to know why. And one of the ways that I think is worth pursuing, and Tamara van der Waal has been really pushing this now, along with many other people, looking at movies and maybe other ways of so not just looking at functional connectivity, but also things like intersubject correlation, so similarity of evoked activity across individuals, as other ways to obtain information related to functional connectivity and look at brain behavior relationships and so on. So I'm going to skip over all this because again, it just highlights this problem of sample size. There's this really interesting paper from Tom Nichols looking at this non-parametric, what do they call non-parametric combination approach to doing brain behavior analyses that I'm quite interested in, that I'd like to follow up a bit more, and I just wanted to highlight it in case anybody is interested in that. So I just wanted to finish by with this kind of problem that I'm struggling with, that we don't always have these enormous samples, right? It's needing these huge samples is a big problem for clinical developmental research, but particularly like for rare disorders. So I'm working with a colleague at Trinity who is an expert in the neurogenetics of autism, and she has a cohort of individuals with neurexin 1 deletions, and at least in Ireland this is pretty rare. She thinks she probably has everyone who has been identified, and that's 34 people, and we have scans on about 15 of those. And to do anything, meaningful with those data, we probably aren't going to be able to do anything particularly meaningful with that small a sample. But I would like to think about, how do we address that? Like, how do we work with these smaller samples in a meaningful and robust and reliable way? And so one way that we can do it is, like I said, we can try to boost the reliability of the measures that we use. We can try to boost effect size. Like, that's the other thing that Sinyan, that Nature Human Behavior paper highlighted. We can boost reliability and we can boost effect size. And so that's, when I was talking about what my lab does, why we're trying to identify these better behavioral assays of maybe group differences. But then the other option, I suppose, is this collaboration, consortia, sharing data, working together across labs, the kinds of things that, again, that this summer school, so in addition to this last thing of findable, accessible, interoperable, reproducible methods that you're learning about and you're implementing and you're going to take with you for the rest of your career, you've also formed this network. And I think that's the other really great thing about these kinds of summer schools, and would always encourage you to do them, because you meet people that, you're all going to go your separate ways. Some of you are all based here in Montreal, but different universities, but you may reconnect again in the future. And those kinds of connections are so important in science. So whether it's to share data or work together in a project, or to get a recommendation letter, or work together in a grant, or whatever it is, these connections that you make here are going to stay with you for the rest of your career. So, and I am more than happy to answer questions by email, because I have to leave today. And if anybody wants to talk about coming to Ireland, let me know. you've probably heard me moan about the poor funding situation there, but there are, opportunities, ways that, especially if somebody's really interested in going to Ireland, we can talk about how we could kind of look at the grants that are available and stuff. But okay, have enjoyed the rest of this summer school. Like I said, I think it's such an awesome opportunity. I want to really thank Pierre for inviting me and giving me this opportunity to come and spend time with you guys. And I learned, I think, just as much as you did from the whole thing. And best of luck with the last 2 weeks.
Thanks,
Thank
Claire.
you.
Well, thanks so much for joining, Claire. As she said, unfortunately you have to go, and the plane is early this afternoon, but you still have a few hours to catch her if you have discussions you want to have with her. Is there any pressing question? Those slides. We've covered a lot of ground here.
Yeah, sorry, I didn't talk
No, but
too
it's
much,
awesome.
But I suppose I'm worried that I think a lot of it's very basic, and you've probably covered it already. So, and hopefully I've just kind of maybe dotted some potential things that you might follow up in a couple of weeks when you have some
the
more
reliability
headspace.
things is very real, and now some people are doing those fairly clean sort of analysis on large sample size and realizing, as you said, that there's little effects, and that's scary. And you were highlighting the issue of the kids with less reliability. We recently completed this work where we have this Simon subject who traveled to Canada, and we were able to do a little bit what Emily Finn did, try to guess who's who based on 2 scans. And when we use 2 scans from the same site, then everything's good.
Yeah.
But when we start comparing a scan from say Philips with Siemens, and things start to fall apart. So it's not just kids, it's also pulling scanners across the country. When you want to scale up things and go in large studies for large clinical trials, actually FMRI, I don't think it's
That's
there.
pretty worrying now. If you've got the same person in different scanners and you're not, the ID is not working.
So within scan, we try to identify him compared to Chinese participants.
Yeah.
It was,
That's probably
easy,
and
we get almost 100% identifiability. When we go between vendors, we go as low as 0.5.
Wow.
So we divide our identifiability rate by 2.
We were trying it with kids and we were getting 0.1. Which is, now I have to kind of caution that was a master's student who didn't really know what he was doing and then dropped the project after about a week, so it's possible that it's all wrong. But that was, so it's something I really want to get back to, but I and it's this thing is really bugging me, how do we get better measures of functional connectivity? And that doesn't mean fancier or more sophisticated or with nonlinear